What is polypeptide synthesis and how does it work? Let's break it down. What is a polypeptide? Poly stemming from Greek meaning many and peptide meaning peptide. A peptide is a chemical compound, two substances that are chemically bonded together. These substances in particular are amino acids, the primary ingredient in creating proteins. What is meant by synthesis? Synthesis simply means the creation or production of something, often from some key building blocks. Polypeptide synthesis, therefore, is the creation of polypeptides, and more specifically, polypeptide chains. There are two primary pieces to the polypeptide synthesis puzzle: transcription and translation. Transcription is the process of single-stranded mRNA receiving information from a single strand of DNA. Translation then involves the mRNA traveling to a ribosome outside of the cell nucleus and creating a polypeptide chain, using the instructions from the specific section of DNA. To get more specific, helicase, an enzyme, breaks the hydrogen bonds between the nucleotides of a specific section of DNA called a gene. This gene that encodes a protein is then encoded onto a template molecule called messenger RNA, or mRNA. This conversion is carried out by RNA polymerase, an enzyme. This process is similar to what happens in DNA replication. However, instead of encoding a new strand of DNA, we are encoding a strand of RNA. Because of this, uracil is used instead of thymine on the RNA side. So wherever is opposite adenine on the DNA, you'll find uracil instead of thymine. The specific order that the nucleotides are in determines the protein created. The mRNA is exported from the cell nucleus via nuclear pores in the nucleus's membrane to the cytoplasm of the cell for translation to occur at a ribosome. Translation is the process of the ribosome creating a polypeptide chain connected via peptide bonds, which will eventually create a polypeptide. Once the mRNA reaches the ribosome, translation begins. Sitting in the cytoplasm of a cell are tRNA molecules, or transfer RNA molecules. These have a singular amino acid at one end, and at the other end it has three nucleotide bases, called an anticodon, which can connect to the nucleotide bases on the mRNA. The ribosome reads the mRNA 3 bases, or codons, at a time, and gives it the complementary tRNA molecule. Then, the tRNA's amino acid is extracted and connected with a polypeptide bond to the next one in the chain, creating a polypeptide chain. These chains form proteins, which, depending on the gene that encoded the mRNA, could be enzymes, antibodies, hormones, transport proteins, and other functions that proteins serve in the body. To summarize, polypeptide synthesis is how DNA, and more specifically genes, have the code to create proteins in the body. This is done through messenger RNA, which transfers the information from the DNA within a cell nucleus to a ribosome within the cytoplasm. The ribosome uses tRNA to gather the amino acids needed for a polypeptide chain. And that's polypeptide synthesis.